So to summarise, they asked for the setup, conduct and reporting of all three of these studies. We have 36 volunteers in total. If they all ran sequentially study to study, it would be around 21 months. So as I mentioned, we thought, well, what can we do with this? There must be a better way to run this all for them. So in the optimized design, you can see we have a combined cocktail study. So we thought, why have two separate cocktail studies when you can utilize the IMP drug administration steady state period once? The longest cocktail study was day 11. However, when you combine them, it only goes out by 2 days. It's only day 13 that it's been extended by. And this is simply by just adding in a 1-day cocktail period for the SIP cocktail, and then again, another day for the SIP cocktail. And it can be this short just based off the half-lives of these probes. They're quite short, and we've only got around a 24-hour PK collection period. Due to the fact that the IMP was the object in the third study part that they asked for, we couldn't optimize this into one big design, so we did keep them separate. However, it's a multi-part protocol, so this design was exactly the same. Kept it in 2 groups of 12 volunteers, but it was ran in one protocol, but 2 different study parts, meaning they could run in parallel, which just streamlines the timelines a little bit. So in this optimized approach, as I mentioned, it's one study, just 2 different parts, running in parallel. We've reduced the subject number, we've reduced the setup costs, we've reduced the setup time and time to reporting because they can run in parallel, which means significant cost and time savings overall. So in this sort of design, we're probably looking at around the 6-month range from protocol writing all the way through to reporting to get information on, I think, about 10 different probes from a cocktail study, and then the CYP1A2 inhibition study. So to summarise, DDIs hopefully I've proved to you that are an essential part of the regulatory clinical pharmacology package. There's multiple ways to get to these stages, and there's multiple tools like the PBPK modelling, and then also all of the in vitro assessments to highlight the risks and where DDIs might be needed. And all of this helps build a robust data package, ensuring that drugs are delivered to the market safely for the patients. And at Quotient, we're able to consult, design and deliver all of these types of DDI studies for your molecules.